So I'll just do like a super brief introduction. Very excited to introduce Allon Klein, who is also at Harvard and does beautiful work on self-weight differentiation. And yeah, excited to hear the talk.
Vanessa, thank you. Yeah, that's great. Let me see. I might put this down to 1, and then I think it'll be fine. Okay, great. So I feel slightly guilty having a title like this in a morphometry workshop, but there'll be sort of tidbits which actually relate to morphogenesis along the way. You just have to pay attention and not fall asleep, and otherwise you'll miss them. So let me see. So let's see if this is okay. So here's what I was going to do today. I'm going to, we had a really nice talk yesterday from Paul Francois about Wellington landscapes. I'm going to start off with a background, which is a brief recap on learning dynamics from single cell snapshots. I'm going to make a little bit of an allusion there to some work on developmental biology. And then I'm going to talk about learning stochastic state transition rates from clonal dynamics. But actually, what I'm really going to do is also introduce a new technology that we've been developing in the lab during that process. And I'm going to also have a brief shout out to some work that Sean will be talking about, which is using this technology along with some other innovations he's made to look at cell adhesion interactions. So those are the ways that morphogenesis is going to be briefly mentioned in my talk, but most of the time we're going to talk about differentiation and dynamics. And then if I have time at the end, Olivier and I, a couple of years ago, were involved in co-founding a company, and we're using some of this technology. And if there's time, I'll just show you a little bit about what we're doing with it, because it relates to trying to learn developmental dynamics as well. So we'll see, if depending on how I control this. Okay, so the question, in a way, there's this. I think that this conference captures a way that mathematical thinking sort of applies to biology, which is that there are descriptive aspects to biology, and then there's mechanistic aspects. And actually, Paul also gave this example of like thinking about Copernican view and sort of heliocentric versus other views. So the first thing that we really need to do in biology is just describe what happens. And some of the earliest approaches to describing dynamics is to label a piece of tissue and just ask where it ends up later on. And this is these are all watercolor drawings from a 300-page paper from Walter Vogt, 1929. Where he took agar pads soaked in different dyes, put them on the outside of an amphibian embryo, and then as it underwent gastrulation and morphogenesis, he could track where the different patches of dye ended up. And this was the very first way of tracking where a cell starts off, in which state it starts off, or which position over here. This is the only piece of information that one has, and then where it ends up at a later point. This is Walter Vogt's work. Oh, this I believe was I think it's a frog. Yes, yeah. This is a Xenopus at the time. Okay. No. Sorry, it wasn't Xenopus. It was another frog. Okay, all right.
I mean, they were collecting.
Right, they were collected from a pond somewhere. Yeah. Okay. All right. Yeah. Right. Okay. So now, fast forward 100 years or so. And we do have other ways of looking. I mean, there's a beautiful light sheet microscopy, for example, and other approaches. One other way of reconstructing dynamics is by taking snapshots and then walking along those snapshots and from there learning dynamics. And the basic principle is that one starts off with a static snapshot, which is a high-dimensional set of observations. Every column over here is one cell, which has been sampled, and a large number of features. And if it was a three-dimensional set of features, we could imagine just looking at the point cloud. In practice, we have a much higher dimensional set of features. Paul talked a little bit about the pitfalls of projecting that down into lower dimensions using methods like UMaps. But one doesn't have to project it into low dimensions. One can try to learn what the structure of these distributions of states are or observations are in a high-dimensional space, and then from there start to learn the dynamics. Now, there's still many, many choices that can be made. For example, there's a distance metric which has to be imposed in order to build such sort of relationships. That means putting certain weight on certain features or sets of features more than others. So this is not by any means it's not like space where we know that that there's an isotropy between different directions. Over here, we have to decide how to weight every dimension. but nonetheless, we can go through a process of building these dynamics. And just to sort of define what we mean by state over here, I'm going to talk a lot about state, so just to get definitions out of the way. You can imagine defining a state of a cell, not only in terms of what's happening inside the cell, but also all of its surrounding. Its tissue context, its mechanical environment. This will predict not only what it's sensing at the moment that you're looking at it, but also what it might sense in the future. So in a sense, it does encode, if you were, say, trying to come up with a Markovian description, knowing what's happening around the cell is important in order to make some statement about what it will do later. Then inside the cell, there's a lot that can be said about the structure of the cell. I don't have over here, for example, pictures of cytoskeleton organization or apical basal polarity. There's a lot more that can be said about cell organization. There's also molecular organization over here. For example, the DNA can be in active or inactive states, which is governed by modifications to histones or methylation of the DNA, which will silence and activate genes. And finally, there's gene expression, which is just the levels of mRNA of different genes. And in terms of very practical readouts, often gene expression is the most mature and easy currency for us to access. So we end up looking at cell state in terms of gene expression, but really there's all of this other information which we hope will correlate with gene expression, but there may be latent information which we can't get directly from gene expression. So when we talk about state, I'll often be using gene expression as a proxy for state, but there's a much more complete view. Okay, here's just an example of these high-dimensional graphs, and I think you've seen many of these. This is just a very, very simple case. It's a case of a self-renewing tissue, so there's very, very little going on over here. You have this is all taking a single basal layer of a stratified epithelium. So these cells will divide when they're in the basal layer, and then as they differentiate, they move upwards. This is a tissue that I've my first introduction to biology and as a PhD student. And you can see very simply in sort of these, this is a sampling. If we ignore these side clusters, which are showing us some sort of different parts of the biology, there's two major events occurring over here. The first one is that there's a loop, and that's the cell cycle. And then the second one is a decision to differentiate, and that's the expression of a gene which is associated with differentiation, and these cells will very soon leave into the suprabasal layers. And as a side comment, you can see that the cells, when they're up here, these are genes associated with being in a later stage of the cell cycle, so S and G2, and the cells can commit to differentiate at this point so that when they relax back down again into G1, they're already doing so during differentiation. So there's this decoupling of the cell cycle and differentiation. The two things are happening, can occur independently of each other, and they can overlap. Okay, just an example. If you now think about an embryo, this is actually a useful framework to sample very rapidly tissues in the embryo. Here's an example looking, for example, at a zebrafish embryo, and this is just pictorially evaluating a series of single cell of clusters, which we can identify as different tissues, which we can, by sampling, simultaneously look at their gene expression, look at the frequency of these cells. And then as the embryo matures, we get a gradual elaboration in the number of tissues. And this is very useful both in order to generate reference maps, just what are the states in the embryo, what genes are expressed, what so signaling molecules or receptors, and there's many other pieces of information which are useful. It also provides a reference for perturbations. If we now are going to start to go and change change a molecular process, how do the frequencies of different tissues change? And we can do this now, not by microscopy, but looking at all of these different tissues at once in one go. So, and this is one taste. This is work that Kalki in my lab did, where we were looking at the effect of inhibiting cell division. So this is one of those few cases I'll talk about developmental biology here. And there's a lot that's been said about the cells moving or dividing and proliferation being important. In fact, growth is very important, but it turns out that if one stops cell division entirely, over here, we use a couple of different approaches. I'm just highlighting one, which is using a mutant, which causes a defect that stops the cells from being able to divide. And now each one of these points is a different embryonic cell state. It's 24 hours of development of the zebrafish embryo. And we look at the frequency of those states in the wild type, and then there's far fewer cells, but once we normalize for the frequency frequency of the cells, we can look at the abundance of the different tissues in the mutant. We see that there's very, very little change in the distribution of cell states across the embryo. It's really remarkably conserved. This embryo is essentially going through the same whole process of development and differentiation. There's a very small quantitative shift in the rate of development, but by and large, you generate all of the different tissues. And you can see this, sometimes seeing is believing. You can see this if you look at, say, this is a single cell layer. This is the floor plate. It's a very, very fine layer. It's stained over here with an RNA stain. And when you block cell division, you still find that this layer of cells is forming. It's just that the cells are much bigger and now they're spreading out more. And the same thing goes here is another starts off as a very rare cell population. It's the pancreatic primordium. It will give rise later to the pancreas. And you can again see that those cells are there. Over here, this is the red blood cell. This is a zone which will give rise to red blood cells. So this is hemoglobin expression. And again, you just get much larger cells, but they're all there forming red blood cells. These are leukocytes and they're fast and slow muscle cells coming from the somites. So this is an example where we can go and stain for each one of these individually, but we can also get a single snapshot very quickly of the changes and the requirements for cell division. Yeah.
Is it possible to do this in a spatially heterogeneous way, or this is right now global?
So you can use spatial transcriptomic approaches.
No, I mean growth control.
Oh, growth control. Can you? I wouldn't. Let's think about this. Would you be able to? I would have to think about it. I think that the mutant that we're using is a germline mutant. But the question, and it's not a gain of function, it's a loss of function. The question is, could you, for example, induce the mutant only in use an optogenetic approach? And the answer I would have to think, certainly this genetic system by itself wouldn't be simple to use that way, but there might be other ones which could be used. Yeah. Okay. All right, so I'm going to now, this is for historical purposes, this is stuff that we've done in the past. I'm going to briefly mention this. Again, this is relating back to Paul's talk yesterday. So we can just use this as a census. If we wanted to learn dynamics from this process, to try to learn regulation, I'm just going to make a simple point, which is that all the snapshots that we see can be related to dynamics through a bookkeeping equation, okay, which is the flux balance equation. The concentration of cells in the high dimensional gene expression space is just the divergence of the flux and net growth and loss from a region. So growth would just be self-renewal or division, and then a loss would be that the cells are being physically removed or dying, depending on the precise details of the experiment. And because of this, learning dynamics from this type of data is always underdetermined because it's only indirectly seen through this divergence. And that means that the learning the velocity is always underdetermined. In practice, many common assumptions are made. So Paul yesterday spoke about using gradient descent as a very strong constraint. As soon as one imposes a gradient, one makes some progress towards simplifying this problem. It doesn't fully simplify because you can have diffusion, and the diffusion is not specified. But that does lead to some simplifications. We had a discussion yesterday about whether we treat the landscapes as frozen or varying. There's also equivalent formulations. So there's actually a huge body of machine learning that has come up using optimal transport. And when one uses an entropy regularized optimal transport, the problem is equivalent to a gradient descent. So it's the same problem. This is just a Schrödinger bridge. These are all different fields coming into the same fundamental description of the same set of assumptions to try to extract what the velocity fields are. And then there's, the different approaches using graph Laplacians, neural ODs or SDs, flow matching approaches, and so on.
But this assumes that you know the final stage to which you want to get. If you're going to use any of these optimal transport.
So often what happens is that there will be a time series, such as the one I showed for the embryo, and now the optimal transport is essentially trying to interpolate between those two states and asking what is the potential gradient which would instantaneously have to exist for the balls to roll down, and one has to make assumptions about net expansion and growth.
So if one did this and between, let's say, time t equal to 0 and t equal to big T, even though you had intermediate snapshots, you did it in one shot, and you did it with 6 intermediate steps, do you get any correlation?
No, that would work very poorly. So typically, there's going to be some might call it, to use a terminology from a colleague of ours, a Nyquist limit, in which you have enough resolution that you can basically, anytime that you take another intermediate snapshot, you're just passing right through it. But if your steps are big enough, you're going to lose some interesting structure. Yeah.
And do you have a sense as to what the optimal sampling is?
If you're looking for a time scale, I think it's going to depend enormously on the biological process and even the organism. I mean, zebrafish develop in 72 hours to something that can hatch. And then in a mouse, it's 21 days, and in human, it's months. So, and then for different tissues, there's going to be very rapid transitions. And it's, so I'm copping out over here. But,
no, I guess my question is, early in development, presumably you need to sample more frequently. Later in development, you better sample.
So my guess is that there are
a lot of decisions.
So it's certainly true that for a mouse, for example, around the period of gastrulation, there's an enormous, very, very rapid dynamics and also a lot of growth. After that, there are tissues which will undergo rapid discontinuous transition. So you get these sort of slow periods and then rapid periods. And where those happen is going to be dependent on the details of the tissues.
Is there any tissue where that is experimentally been mapped out as a clue to figure out how best to sample?
You know, there's a beautiful data set that's come out of Jay Shinduri's lab where they've taken the whole mouse embryo and sampled it at approximately 1 hour resolution.
Through all gestation?
Through 21 days. Now, because there's about 500 million cells by the time a mouse is born, the later stages are enormously undersampled, and you only get the very abundant tissues at that point. At the early stages, I think you could reasonably answer how many time points can you drop out and still do a good job. Yeah. I don't know the answer, though. In principle, it's addressable. Yeah. This is just sort of, I don't want to spend too long on it. This is an example of sort of the recent games that people play. So this is reformulating the problem of trying to learn dynamics using a stochastic differential, neural stochastic differential equations. Over here, we have a drift term and a diffusion term. We treat both of them as state dependent. So, you know, essentially at the next time step, we have a drift and a diffusion. And the drift and diffusion are both functions of the local state of the cell in some embedding space, but it's a nonlinear function. And therefore, there's a neural network which maps the state to the velocity vector. And then the goal is to try to learn both the diffusion and the drift. And in this particular formulation, the diffusion was treated as inhomogeneous and anisotropic. So that leads there's quite a substantial number of degrees of freedom there. So this requires very strong regularization. So it's more and more assumptions have to be added to this process in order to make the curves, to come up with any curves at all. I'm not going to spend too long on this. I want to just point out that with all of this information, one can still be misled by snapshots. And these are some cartoons to just illustrate it. You can imagine that you look at one of these high-dimensional spaces, and there's a bifurcation, and you say, okay, the cells haven't decided what they're doing until they get to this point, and then they take a decision left or right. But of course, topologically, this seems like it's exactly the same problem, but it could be that the cells are making the decision far earlier. And of course, in terms of what matters to control these cells or to understand the decision-making process, the state of these cells is quite different from the state down here. So this is, whether they're committed or not is quite important. And this degree of freedom may be completely determining whether they're committed or not. And that would be very important to know. There could also be convergence. You see this actually neural differentiation. It looks like a bow tie. The cells initially in the spinal cord are presented in different layers. And then as they go through a neural maturation differentiation, they have a very strong transcriptional signature which really wipes out their spatial location in terms of gene expression. And it looks like they're in this common state and then they split. But in fact, they're completely predetermined. They come from very different layers. They've been separated for a while now. So this is sort of an extreme version. Yes.
Can I ask something?
Yeah.
So I find this problem extremely interesting because in a classical way, these sort of differences have been teased apart with very mechanical experiments, right? Transplantations and checks of fate and commitment, and at what time can you put a cell in a different area of the embryo, and so on and so forth. But these experiments are not possible in most systems, and so I was wondering, what's the state of the art in trying to
Yeah.
make these differences from transcription profiles alone?
So I think I'm going to give a different answer, which is just this one over here, which is one way to try to constrain the problem further is with lineage tracing. And I think that transcription alone, I think that so one answer to that is spatial transcriptomics, because one can actually get a very clear view that cells that are seemingly in the same transcriptional state are actually in different spatial states. And there may be transcriptional signatures there that weren't paid attention to, which one can then learn and obtain information from. The other approach is to try to build in lineage information so that we can now find which cells are related to which cells. So this is, in some cases, you can really do this with an experimental manipulation. This is not general to development. So in this particular case, cells are barcoded ex vivo, and then some of them are transplanted back into animals, and their sister cells are sampled. And now we can relate the early state of a cell to its future fate and identify fate commitment points. So this is not something, again, I'm putting this in the spirit of background, but I think that spatial and lineage information helps overcome a situation where we have to rely only on transcriptomes alone to answer this question. Does that,
But
does that help?
can I just make another, because I was more thinking about the fact that lineage tracing is one way, but it still doesn't tell you if a cell is committed to a fate or if it can still change fate, right? And formally, no, formally you test that by taking that cell, put it somewhere else and see if it changes fate then. And so I was wondering, I am a bit ignorant of the recent advances, if people are thinking about how to try and make those sort of tests, or if there's any way to.
Okay, so I think, you know, okay, one answer to this is as follows. In many of the cases where people weren't sure whether the cells are committed or not, when you look at the transcriptomes of the cells, it's very evident that they're strongly committed. So in some sense, this is just pushing the problem back a little bit. It's just saying that many of the original questions, is this field of cells uniform or not? It just, those problems have just completely gone away because it's very clear that those are not uniform fields of cells. And, you know, for one example, this is when we were looking at the Xenopus embryos, there were many cells which in the literature were reported to appear at a particular time in development, and we could identify that they appeared several hours earlier.
A lot earlier.
So that's an example of a situation where, you know, you would have done a commitment experiment, but in fact, we can just see that all of the transcription factors associated with that cell are all very, and all of the some early downstream effectors are all there already, and it would be surprising if those cells weren't committed. But I think that beyond that, there's probably no magic to be done.
I think what she is asking is also in the notion of commitment, there is this notion of irreversibility. Which
I don't know if there's essentially, is there a universal signature of irreversibility? And I don't know, I'm not aware of that being the case. So can you just look at a cell and say from transcriptomes that it's irreversible? I don't think I know the answer to that. I mean, there's certainly there are very, very clear examples of cells that look very differentiated, but they're plastic and you know, in the intestine, for example, and they can be reverted back. And so I don't think that there's a universal answer.
Are there experiments which show that the time required or the perturbation size required to reverse is a function of how far along you are in the path? So sort of in the context of first order transitions, for example, as you come closer and closer to a critical point, everything becomes slower and slower. Are there experiments which are suggestive of that, even at a population level?
If there are, there would be in vitro. And, you know, equivalent to, say, cases where one withdraws a factor and then asks about the, I don't know, I think we can maybe, I'll tell you what my thoughts later. I think there's one or two papers, but I don't think I know enough to be useful to an audience right now. Okay. All right. I'm going to just skip this in the interest of time. I'll just say that maybe I'm just going to skip this, actually. I'll make one point. So when one looks at what individual cells are doing, it often appears, when labels a cell and follows its fate, it can often appear stochastic. So essentially it looks like it's a probabilistic decision. And this can lead over multiple cell divisions to branching processes, which can be very complex. So you can get large distributions of outcomes. But in fact, and I think that particularly for this audience, it's interesting because morphogenesis and morphometry is often thought about at the level of a tissue rather than the level of individual cell. And in fact, these highly variable behaviors at the level of individual cells can come about through very, very simple rules at the level of a population. So this is an example which we understand very well, which is if you have a monolayer, this is like the epidermis, during self-renewal, there are cells that are pulled out to supply the suprabasal layers. This is the same example of a tissue that I showed earlier. And the response of that is that a neighboring cell will divide. And this process is just very, very simple. You take out a cell, you divide, you take out a cell, you divide. But the result of that is that for any individual cell, that process will lead to what seemingly looks like a random process. Formally, this is actually going to be a process in which there's cell-cell interactions because one cell is responding to its neighbors. But in 2 dimensions and above, this will effectively look like a non-interacting system. So it'll look like a random process. Okay. So it's a little bit about, the sort of single cell RNA-seq 101, a bit about dynamics. Here's the problem. In the spirit of this being a research talk, I'm going to sort of now transition maybe from background to thinking about dynamics. And the problem that I'm going to talk about today is really about dynamics. We've talked about the fact that just learning from snapshots is an underdetermined problem. What we'd like to do is be in a situation where we have cells in a particular state and we can measure the rate at which they transition to another state. And we'd like to ideally do this for all states over time. And again, in the spirit, this is sort of like Paul Francois. We can think about this as maybe cells in one state, being trapped and then transitioning to another. What we actually have, if we look at, say, lineage data, is that we have clones or we have some dynamics. And we can either do optimal transport, move our entire distribution over, or if we actually have real lineage information, then what we're ending up with is just an observation of the distribution of states at the end. And we now are trying to infer from these distributions the rate of switching that's occurring in that population. So the question is, the question I'm going to address is this inverse problem. How do we go from a collection of observations of a clonal state to the rates of transition inside a clone? And I'm going to do this in a very simple context, which for this audience will be forgiven. Okay, so that's basically going to be the next part of the talk. Okay. All right. So I'm going to introduce for this a very simple analytical idea, which came from Arjun Raj. He's at University of Pennsylvania, which was a very simple idea called memory-seq. He called it at the time. He took single cells, he put them into 48 different wells and let them grow. And he had this idea that if you have rapid switching, then the memory of the initial state of the cell will rapidly be lost, and you end up with an average expression, sort of central limit theorem. Whereas if you have very slow transitions, then some of these cells will stay in a particular state, and now you will be able to see that the heterogeneity doesn't subside as these clones are growing. So you don't converge onto an average. And the idea is that from here, you can just pick out switching rates. Right?
Yeah. What do you mean by transitions?
So over here you have some cell there's some program. It's either in a low state or in a high state. The cells can switch. If you happen to capture the cell in a high state, does it stay in a high state? If you happen to capture the cell in a low state, does it stay in a low state? There's some average occupation. This could be at the level of a program. It could also address this at the level of an individual gene or any other feature. Okay, so what they did this at the level of, they grew these 48 colonies, and from here they identified, this was in cancer cells, they found a series of genes which were persistently expressed in a subset of cells, and they actually then proposed that those might be important for cancer resistance, and they followed that. So that was their study. And in our lab, I'm going to talk about some work that Ignas has done. He's a graduate student in the lab to try to think about how we can generate those kind of data sets at scale. So now I'm going to transition a little bit to a technology story. And in order to address this problem, what Ignas did was he developed these, adopted a technology which had been maturing in the soft matter community. And this is using a core shell capsule in order to generate very large numbers of colonies. So what we can do is put a cell inside one of these capsules. I'll talk a little bit about them in a minute, and then grow the cells. And once the cells have grown, we can then extract DNA and RNA from each one of these colonies and analyze the state of the clone at that point. Now, in order to do this, we need a semi-permeable compartment so that we can happily grow these cells. They'll freely exchange media. And we also need to be able to then later on add in reagents to do molecular biology analysis. But we have to trap the cells inside. And we also then later on want to trap the DNA and RNA. So in order to do this, there's been a whole field of soft matter on making these core shell capsules. And we played around with this in order to get a very good composition so that we could get very sharp size cutoffs, which would allow us to retain not only cells, but also molecules of the right size inside these, okay? This is an idea, the idea of using these types of microcompartments for large-scale genomic assays is something that was proposed initially by a colleague of mine, Linus. He and I worked together in our postdoc, and we've been collaborating since, and it required a little bit of working out. So I'll tell you a little bit about this. So what we've come up with is a water-oil system. This is a droplet microfluidic system, which then uses liquid-liquid phase separation to separate an outer polymer from an inner polymer. The outer polymer then undergoes cross-linking. We can then wash away the oil, and we end up with these compartments. We can generate millions of these per hour. They're very, very simple to make. And the key is, in order to have an outside shell that is reliably wetting the exterior, we use an amphiphilic polymer so that it likes to sit next to the oil phase and it goes to the outside. And these are porous, and there's a lot of tuning went into just getting the porosity right. I'll show you a little bit about that. This is just a cryo EM image just to show you this is the outer shell. There's also an inner polymer, which is driving the phase separation. So you get these sort of two phases over there. We can digest away the inner polymer, and we do that for many of the experiments. And then there's some porosity to this process. Just to show you the porosity, so if you have a short DNA molecule, these will diffuse out. If you have a longer one, they'll stay trapped inside. We can measure the diffusion, and then just as a fun observation, it turns out that these behave like a porous media. So the diffusion of molecules out of this follows what seems to be a power law relaxation. If anyone is interested in that, you can.
What sets out?
We're not sure yet. Yeah, I mean, we know that the size of the molecule that we're diffusing out sets alpha, but what properties of the capsule set alpha, we're not quite sure yet. Yeah,
How much control do you need for it to be porous to certain things?
So what was very important for us is just very practically, we wanted RNA to stay inside, and that's about 500 nucleotides or longer. And we wanted oligonucleotides of 100 nucleotides or less to freely exchange. And that's a pretty narrow range. I mean, it's over a 5-fold difference. We wanted a fairly sharp cutoff. But it turns out you can achieve this reasonably well. And I think you can see that over here. This is on a log scale. 100 nucleotides will diffuse within order of tens of minutes. And by the time you're at 300 nucleotides, you're at, it's like 2 orders of magnitude more almost. Yeah. Yeah. So this is sufficient for us. 1 kilobase of double-stranded DNA never leaves, basically. It's trapped inside. Yeah. Okay. So we can also grow cells inside these. Maha, this one's for you. That's an iPSC that's developed into a blastocyst right there and trapped inside. I know you've looked at those. We can grow primary cells like hematopoietic stem cells. We've grown lots of different cell lines in here as well. They start off with 1 cell and they expand. Sean will be talking a little bit about his work. He's using these actually in a purely utilitarian way. Really, the emphasis of his project is not the cages themselves, but the cells. He's been generating libraries of optically barcoded cell lines with different coherence, and then we can generate thousands of these different colonies and look at the way that the coherence sorts and interact with each other. And each one of these now has, we can look at all the pairs, but each one of these starts off with a handful of sort of Poisson distribution of say 4 or so different cells. So you also get triplets and quadruplets, and you can see all of the interactions between the different coherents. From here, you can start to look for, the hope is that we can order the relative interaction strengths, but also look at changes in cell morphology that are, you can start to see tightly compacted cells, and then there's cells that are sort of more marble-like and so on. So this is, Sean will tell you about this. I won't, unfortunately, tell you. I would have told you about this if Sean wasn't.
Can I ask a question?
Yeah.
They luminize at all, or they remain?
These cells don't luminize. Yeah. Yeah, they don't. Yeah, this particular cell line. The library could be put into another cell line. Yeah. Okay. Yeah. Okay. So we can do that. These multi-step genomic assays, and I'll just briefly tell you now. You can imagine that we can now take this memory-seek principle and look for cells that look for genes that are, changes in expression, but also the persistence of gene expression, right? So these are very persistent because the colonies will stay in stable states, and this will be sort of low persistence. Okay. All right, so you got this kind of data. There's about 130,000 different cages over here. These are expanding clones over, 2, 4, 6 days. And this is from one particular cancer cell line that we looked at. And you can see that there's quite a bit of variation. Each point over here is not a cell, it's a colony. And you can see that these colonies are not all collapsing into a single blob. There's some structure to this data. You can see quite a bit of persistence in heterogeneity. Here's another way of looking at it. So if you just look at variable genes, so this is a gene-gene correlation matrix. If you were to randomize, take single cell data from the same cells and just sum them together, this is central limit. All the correlations break down, whereas inside the clones you see a lot of structure remaining. And from here you can now use various approaches to pull out coherent programs, which are these persistent programs that last over some time, and we can define a series of these programs. This particular is an erythroleukemia, so you can see hemoglobin is expressed, for example, in one of these programs. I'll come back to that. And just to sort of convince ourselves we're not imagining things, we can now take these and stain them for different factors. So here's a more mesenchymal program, sort of vimentin. Here's a hemoglobin program, and then here's some sort of double positives. And we can sort of also look in our data at the distribution. Most of the colonies are either one state or the other, but there's a few that are in this double positive state.
Do you control the concentration of one to the other?
So no, so this is just stochastic variation in the population, right? We're just observing these. So these cells exist in one state or another, and we can now just observe. Yeah, this is just an observation. Now, what we want to do is to learn the rates of switching. So the way that we approach this is quite simple. We can think about essentially the probability of a clone being in a particular state, which is the distribution of its cells in a particular state, is some Markov process, the chemical master equation, which includes, yeah.
How many states?
So in this particular case, you can marginalize this and just think of every program being on or off, and there's about 15 meaningful programs, or 12 or something. You can also think about the joint distribution, right? So you can think about switching from one state being high and another state being low to flipping the other way.
Because here, when you're going to look at the rate.
Yes. So over here, we're just marginalizing. We're just asking, does the program stay on or off? And we're looking one state at a time. There is a higher dimensional version of this, but we've just kept it simple for now. And we need to take account of two things. There's growth inside the clones, and then there's each one of these cells can now independently switch with some rate. So this is a very simple model. And from this, we now want to take our distribution of the usage of a program across all the clones and over a time series, try to fit the switching rate. So there's a forward rate and there's sort of on rate and an off rate, and try to learn those rates in order to fit the data. The way that we do that, and there's growth in this process, I suspect that for the very simplest example over here, there's just a perfectly analytical solution, but just to, in the age of machine learning, we've taken the following approach. We just simulate a very large number of parameters, conditions, simulate them, we look at the distributions, and then we train a surrogate model to essentially predict as a function of parameters what is the clone size distribution that we get. And now we just have essentially a fitted version of the solution to the chemical master equation. So we don't have to solve the chemical master equation. We can plug in any set of parameters and we can just get out an interpolation. And we do this responsibly. And then from there, we can now go through a process of curve fitting and learn the rates. So I'll just show you sort of an example. This is an example of one of these programs. So you can see that most of the colonies don't have the program expressed. And then there's a number of these colonies which do have the program expressed, and you can now fit, and you get a sort of a very, very low on rate and some rate of off rate, right? And that's an example of learning one of these sets of switching rates from this program. And yeah.
You said very small and very large. Can you elaborate a little bit?
Yeah, so this is per cell cycle.
Yeah.
So this is saying that about once every 4 cell cycles, you will deactivate the program, but only, say, once in every 100 cell cycles, you'll switch it on.
Why there is such a time scale separation?
So there's 12 programs, and each one of these will give me a different set of these numbers.
Oh, so there's enormous variability.
Yeah. So
the fact that one is 0.3 and the other one is 2 orders of magnitude smaller.
I mean, if you think about it, first of all, I mean, you can sort of eyeball it. The fact that most of our colonies are just off is telling us that the average is low. So that these are rare events, right? Switching on this program in the current growth conditions is rare. Now, actually, this is a very old cell line. It's been studied to death. This is the hemoglobin program, and it's known that, for example, if you add in hemerythrin or other, you can activate this program. And this is equivalent to essentially driving differentiation of this cancer cell. So it's been looked at as this is an example of a situation where the cell is, by and large, staying in the undifferentiated state, which is bad for it as a cancer cell. But, and under these normal growth conditions, which we've chosen, this is a very standard culture, they will very rarely differentiate. I'll show you in a minute that we can drug these cells and then change these rates, right? So this is all tunable.
Yeah. Technical-ish question is, you have to worry about things like large deviations rather than thinking about averages over here because there's such rare events?
So because we have about 100,000 clones to look at, I would say that this is probably not, and there's expansion occurring. If you think about the number of cells in this experiment, by the end of the experiment, there's millions of cells in total. This is actually not a rare event on the scale of the experiment, right? This will happen many times over in the experiment.
You think this is specific to cancer cells, or this is a general?
So this particular formalism is useful for a system which is in steady state. If you were thinking about a dynamic system, I think you would have to just, I think that there's an equivalent formalism to thinking about.
Even IPS.
Yeah, so you can think about exit from pluripotency and then, right.
Or, yeah. No, because cancer cells, they have all kinds of mutations that make this program maybe unstable.
Right.
And I'm just wondering how.
Yeah. I mean, what's baked into this particular model is the idea that it's a memoryless process, that the cells wait. And I think that's the deeper question is whether this model is correct for if you look at the waiting time distribution, it's not exponential, then this model would be a very approximate.
Arjun's work is also in cancer cells.
Yes. Yeah. I think I'm sort of starting to run short on time, so I'll just briefly mention the following. So we went in and we said, look, if this is about, if the phenomena that we're seeing is that there's some states which are persistent, they're very rare, can we change the memory of these cells by drugging their epigenetic state by essentially inhibiting the ability to silence genes, so we can force the genes on? And we do that by, I won't go into the molecular details, but there's a couple of pathways that we play around with. And you can imagine 2 types of changes occurring. So the first one is that we just bias the cells from one state to the other. And then the second one is that we change the persistence, right? Which is a bit like changing the depth of the epigenetic landscape. And now you have more data, right? Because you now have the same dynamics on the program, but now you can look at it under different drug conditions. And this is that same program as before, but now you can see that under these drug conditions, we push the program to switch on, and we slow the program from switching off, right? So you can tune this. And you can now zoom out and ask, well, how is that typical? So that's the program that we looked at. And when we drug the cells in these 3 different drugs, we see that the program tends to switch on. The vast majority of these programs weren't induced to switch on when we inhibited gene silencing. And there's actually a couple of programs which became less prominent when we inhibited gene silencing. And then if you ask about persistence, we're inhibiting the ability of the cell to write memories on DNA, and yet there's no global increase in persistence, right? Some genes, some programs went up and some down. So this is messy. It's not, I mean, we can, the achievement over here in a sense is that we can measure these things, and we can see that if you were particularly interested in one program, you'd get there. If we're looking for a principle, a drugging epigenetic states is going to change the memory of these cells, I don't think that there's any evidence over here. This is sort of a negative result, right? Okay. So I'm going to summarize that, and then, like, you know, I'll overrun by maybe a minute or two to just quickly show you something. So, I think I told you that we can learn rules of cell switching rates from clonal statistics. And in cell lines, these program switching rates are sensitive to drugs, which inhibit epigenetic silencing. We don't simply reduce memory, right? So we see that there's some programs that increase in persistence, other ones that decrease in persistence. But we have now a lens. We can sort of look at this, which was challenging before, because we have this approach, which is high throughput. I also mentioned briefly that there's this technology which we're excited about. There's actually a number of projects in the lab which are now using this, and I think that you might have even more fun hearing what Sean is doing with this, because it's really related to cell adhesion and morphogenesis. But in general, this is a useful tool. These can be generated rapidly and cheaply. They have these sharp permeability cutoffs, which make them very suitable for genomics as a downstream application or readout. We can do these multi-step reactions because we can sequentially wash these capsules and change their conditions. And they open this idea of high throughput live cell assays with genomic readouts. And the example I gave is just a simple one. Okay, so maybe I'll just do this very, very quickly. Olivier and I, together with Cliff Tabin, Jay Shinduri, a few years ago, set up a company that was called Somite, which is appropriate for a company that Olivier is. And then recently we were told that that's not a sexy enough name, so now it's called Cellular Intelligence. The question that we started it by trying to think about learning and predicting developmental transitions for therapeutic purposes. And at some point, we started to think about maybe this technology is useful there. And the challenge is that we can think about starting off in a pluripotent state. And you can see, by the way, the slides have become much more polished. This is from... All right.
That's an AI thing or a CI thing.
It's a CI. That's right. This is CI. It's not AI. But the challenge is you want to end up targeting differentiation into different states, but there's this very large and sparse search space. There's many factors, many steps, many combinations and concentrations. And the question is there a way of efficiently sampling this? And the idea that we came up with is that we would grow iPSCs inside these capsules. And then we could go through a split and pool process in which the cells are exposed to different conditions. They're labeled at every step. And then at the end of this process, we can then go and read out what we get. So it's very, very similar to the type of approach I was showing. But now we're adding in this dynamics of split and pool while the cells are still alive. So this is very much a multi-step readout. And I'll just show you, because this is there's been a lot of work in the company in the last year or year and a half. This was the very, very first proof of concept that a few people in the lab did at the company a while ago, which is just, this is, together with Olivier, this is just doing a 30 by 30 by 30 condition experiment. So that already gives you 27,000 iPSC differentiation protocols, and you generate this very large diversity of cell states at the end of it. Now the company's been doing more, and there's been sort of more sophisticated readouts of what we get at the end. But I just give you so this is this could be an interesting application, and I think it'll be nice to see where this takes us. Okay, all right. So that's it. I'll stop there. I'm a few minutes over. I apologize. Just to say, highlight really Ignace and Sean, and you'll hear more about their work there. There's Ignace. There's Sean right there. And I also wanted to say, if you know anyone, we are recruiting postdocs. If anybody's interested in joining a lab and talking about some of these ideas, and send them my way or come and talk to me yourself. Okay, thanks.